In this video, we're going to talk about final exam review part 3, which is focusing on cell respiration. So let's get started. The first question we're going to take a look at for cell respiration is the different parts of a mitochondrion. So when you're looking at a mitochondrion, we have the outer membrane, which is part A. We have the dog barking. Toby, Akio. All right, let's restart. All right, in this video, we're going to talk about final exam review part 3, which is cell respiration. So the first thing we're going to take a look at is mitochondrion. When you're looking at a mitochondrion, you have different parts of the organelle that allow different reactions to take place during cell respiration. So part A is the outer membrane of the mitochondrion. And as a reminder, mitochondria are examples of the theory of endocrine meiosis. So that's something for you to keep in mind from review part 1. We also have part B, which is the inner membrane. The inner membrane has a lot of folds on it, which allows just a lot of cell respiration to take place. Specifically, our very last step of cell respiration is called the electron transport chain. And the electron transport chain, the molecules that allow the electrons to be transported, and other reactions are all located on the inner membrane. So increasing the amount of membrane, right? You see all these folds, it allows a lot of area for a surface area for the electron transport chain to be located on. And we're going to take a look at C. C is the mitochondrial, I don't know how to say, is the intermembrane space. So it's between the outer membrane and the inner membrane, which is this fold. So the in between intermembrane space. D is called a cristae, C-R-I-S-T-A-E. Cristae are these bumps on the inner membrane, the folds on the inner membrane, we give it a name as well. And then E is the mitochondrial matrix. You are welcome to pause and check your answers over here. So one of the other things that we took a look at, or we're going to take a look at, is the overall chemical equation of cell respiration. So if you know the equation of photosynthesis, you can take it and just flip it, and then add ATP to one end, and then you have the overall chemical equation of cell respiration. Here's my dog deciding to throw the toy. Toby, come here. Hold on a second. Toby, go to bed. And then Marchie's also getting very excited because Marchie loves craisins. Okay, so let's get going. So the next thing we're going to talk about is called anaerobic respiration. So when we say cell respiration, we are often referring to aerobic respiration, but anaerobic respiration is also something that takes place both in our bodies and also other types of organisms. So there are two types of anaerobic respiration. One is called alcohol fermentation, and the other one is called lactic acid fermentation. So both of these anaerobic respiration go through the first step of the aerobic respiration called glycolysis, where you take one glucose, which has, if you see my 6 fingers, is representing the 6 carbons in a glucose. So during glycolysis, this 6 carbon molecule, glucose, is going to split up into 2 pyruvic acid or 2 pyruvates. But during this process, we're also gaining 2 ATP, like a net gain of 2 ATP that can immediately be used inside your bodies. So anaerobic respiration would just go through glycolysis a bunch of times and some other chemical reactions, but it is glycolysis that allows the ATP to be released. And if you just do it, it's just one step, right? You take one molecule, you split it. It's actually more than one step, but overall, it's a pretty simple chemical reaction. So you can go through it pretty fast and just get a bunch of ATP, even though you're not breaking down glucose to its full capacity. It is a nice way to gain ATP when, for example, it's not possible to have oxygen to allow aerobic respiration, or some organisms simply don't do aerobic respiration. Okay, in our bodies, we do something called lactic acid fermentation specifically. So this is when, if you're lifting, you're power lifting, you're assing a lot from your muscles. So your muscles are really strained right now, right? So you need a lot of ATP in a very short amount of time. And also, maybe your heart is pumping very fast already, but the oxygen can't get to your muscles fast enough. So two reasons. One, you need the ATP really fast. And two, maybe you don't have enough oxygen in some of those muscle cells. So your backup method, and you know, the better method is to use lactic acid fermentation in those muscles specifically. So that's how your muscles will start feeling sore after several hours or the next day. So both of these are anaerobic respiration, and lactic acid fermentation is what we do as humans. A little comparison chart over here. Aerobic, air, right, uses oxygen. Anaerobic does not. Efficiency means how well are we really breaking down the glucose. So aerobic respiration breaks it down really well. So we say very efficient. That means we have this amount of available glucose and we can get as many ATP out of it as we possibly can, versus anaerobic respiration is inefficient, as in we're really not using the full capacity of the glucose, but the beauty of it is that it doesn't need oxygen to allow the process to take place, and it is a really fast process. So going to the next one, ATP produced per glucose, 34, 36, 38, somewhere in the 30s, depending on, just depends. Anaerobic respiration is just two ATP produced per glucose. That's why we say it's inefficient. When does it take place? Aerobic respiration in a human body is all the time. Anaerobic respiration is only during very strenuous exercises. Speed of, as in how fast does this chemical reaction take place? Aerobic respiration is slower, and anaerobic respiration is faster because it's just one little step, versus aerobic respiration has a lot of steps involved, which we'll also take a look. Does it go through glycolysis? Yes and yes. And over here, you're welcome to pause and take a look at the answer if you need to. All right, moving on. This is just an overview of aerobic respiration. So I'm going to walk you through the steps on the right side, and then we'll look at some of the details. So step one is glucose going into pyruvate. This is called glycolysis. So we go from this to this. The reason why we do this is because glucose has 6 carbons. As you can see, we have 6 fingers, right? The first step, we're going to break the 6 carbons into 2 3-carbon molecules. So this is one molecule, this is one molecule. These 2 molecules are called pyruvate, which you can see on the screen right here. So if I were to ask you, how many carbons does each pyruvate have? You say 3. How many pyruvates can you produce per glucose? You will say 2, because you have 1 and then another one. Okay. And this process of glycolysis takes place in the cytoplasm for all the cells. Okay, second step, we take the pyruvate. It needs to go from the cytoplasm of the cell into the mitochondrial matrix. So you need to come into the matrix, crossing the outer membrane and the inner membrane. So then our next step is called pyruvate processing. We put one finger down. We're putting one finger down because this 3-carbon molecule will become a 2-carbon molecule called acetyl-CoA. So you end up with 2 acetyl-CoA, 1 and 2, and each acetyl-CoA has 2 carbons. The extra carbon, I don't know why that just happened. Why did these balloons come up? These the extra carbon that got dropped off becomes CO2, carbon dioxide, and you breathe it right out of your body. All right, our next step is called Krebs cycle. So we go Krebs cycle. This Krebs cycle and pyruvate processing both take place in the mitochondrial matrix, which is the very inside of the inner membrane. The very last step, we have the electron transport chain, and we have chemiosmosis. So electron transport chain is very similar to the electron transport chain of photosynthesis, which we'll also take a look, and chemiosmosis is where we are going to make ATP, like a ton of it. Okay, so you can also see how the electron transport chain, this very last step, takes place on the inner membrane of the mitochondria, and this is the step where you're using molecules of oxygen, and water is produced as a byproduct, as in you're not producing water on purpose. You have plenty of water also. It's nice to get a little bit of water, but this is really not enough to sustain you, so you still need to drink a lot of water. Water is just produced as a part of the process. What you really need is to use that oxygen to help you produce a lot of ATP. Something else that you can notice is molecules being transferred from glycolysis and some of the other steps over to the very last step. So we'll take a look at the details of the steps. So right here, we can see the detailed overview of cell respiration. We're going to break it down in the next few slides and talk it over. So very first step, we have our glycolysis. So once again, we're going from glucose to 2-pyruvate, but this is not without a cost. So what we need to do over here is that we actually need to have an input of 2 ATP, a little bit of investment ahead of time. And when you put in 2 ATP during glycolysis, you can make a total of 4 ATP. So you're really gaining 2 ATP, okay? So what is the net gain of ATP during glycolysis? It will be 2. What is the total number of ATP that you're gaining during glycolysis? That will be 4. And how many ATP do we use per glucose? Glucose as a reactant is 2 ATP as well. One other thing that we notice over here is the production of something called NADH. So NADH, just like NADPH, P for photosynthesis, is the electron carrier that we use during photosynthesis. And NADH is also an electron carrier, but now this is the electron carrier for cell respiration. So we have NADH being made using the empty electron carrier without the electrons called NAD+. So while glucose is breaking down, some electrons, high energy electrons, are being released, and NADH is going to grab it and use that energy later. Okay, so all of this is the first step called glycolysis. We have one main reactant, which is glucose. We have 3 products, ATP, so you can say pyruvate first because that's your main product, pyruvate, ATP, and NADH. Second step, we can see how the 3-carbon molecule pyruvate is transferred into the matrix before pyruvate processing takes place. So this is our pyruvate processing. During pyruvate processing, we drop a carbon, right? So 3 carbon goes into 2 carbons, and the 1 extra carbon becomes CO2 and you breathe it out of your body. During this process, NADH is also made. Our fancy little electron carrier is going to carry the electrons to use the energy from the electrons later on. So that's our second step, pyruvate processing. We have one main reactant, which is our pyruvate, and we have 3 products over here as well. Acetyl-CoA, our main product, the 2-carbon molecule, CO2, and also NADH. As you can notice, there is no ATP being produced during this step. Our third step is called Krebs cycle. So the one main reactant is acetyl-CoA, your 2-carbon molecule that was produced during pyruvate processing is going to go into the Krebs cycle. I apologize. So now, during the Krebs cycle, you will make 4 products. We have CO2, ATP, NADH, and FADH2. So all the products that we talked about earlier, they all show up over here. So remember, the first step, glycolysis, allowed you to make ATP, NADH, and pyruvate. And then the second step, we had acetyl-CoA, CO2, and NADH again. Our third step, we have CO2 again, ATP again. Obviously, you can't make glycolysis on acetyl-CoA here because acetyl-CoA is used as a reactant, right? So CO2, ATP, NADH, an electron carrier, and an added on bonus, FADH2 as another electron carrier. So both of these electron carriers will then take the energy from the electron, grab onto it, and then use that energy later on as well. Which brings us to our very last step called the electron transport chain. So during the electron transport chain, it'd be really nice if this was animated. I didn't get the right version of the slides. Okay, so what we see over here, we have the FADH2 and NADH. They're the ones who are electron carriers, but right now they are holding on to the electrons as well. So they'll come over, right, from glycolysis. NADH comes from glycolysis, pyruvate processing, and Krebs cycle, and FADH2 comes from Krebs cycle only. All of those electron carriers will come to the electron transport chain on the inner membrane. You can see the fold on the inner membrane of the mitochondrion. So what they will do is that they will then release the electrons, allow the electrons to go down the electron transport chain, right? These are molecules. The electron transport chain is really made up of molecules on the inner membrane. So they'll take the electron, send it, take the electron, send it over. So while it's While the electrons are going down the electron transport chain, we will then pump the hydrogen just like how we did during photosynthesis. So as the electrons are going down the electron transport chain, we pump hydrogen. And you want to remember that the hydrogen is pumped from the matrix to the intermembrane space. So the intermembrane space, as you can see, the blue dots are the hydrogen ions. You have a lot of it. This is where you have the high concentration of hydrogen ions. They're all very close together. So if you want to put even more hydrogen ions in the intermembrane space, this takes energy, right? So where does that energy come from? We say the electrons go down the electron transport chain and pump. So the energy for the pumping, the movement, the active transport of hydrogen ions from the matrix to the intermembrane space is dependent on the energy from the electrons, which is why it's so crucial for us to understand why are we making the electron carriers earlier, right? CO2 breathed it out. The electron carriers, it's still serving a purpose, a very important purpose, because so far we've only made a few ATP, right? It's really not going to sustain you as a human. So at the end of the electron transport chain, the electrons come out of the electron transport chain and combine with oxygen and form water, okay? And once again, forming water is not the purpose over here. Oxygen combining with the electrons is the purpose. And you ask, why would oxygen care so much about the electrons? Well, they don't. It's just chemicals interacting with each other, but we care about the oxygen because oxygen not only combine with the electrons, it really has such a high affinity for combining with the electrons, as in it really, really, really want to combine with the electrons. Oxygen is just hanging around, craving electrons. Okay, it's electrons hungry. So it wants the electrons so much that it actually allows all the electrons to move through the electron transport chain. It's like a big straw that absorbs the electrons constantly, and that wanting of the electrons is what allows the movement of the electrons. And why would the movement of the electrons be important? Because the movement of the electrons is what pumps the hydrogen. Okay, and then we'll talk about why is hydrogen important, okay? But really right here, it's really important for you to know that oxygen is what we call the final electron acceptor, because it's combining with the electrons at the very end of the electron transport chain, the final electron acceptor, accepting the electrons, okay? And also, what else? The electron donors, right? The source of the electrons are NADH and FADH2. So it's good for you to have this image in your head on how the processes work. All right, our very last step of the electron transport chain, after it's all done, the hydrogen ions will flow through this big purple molecule. It's called the ATP synthase. So as the hydrogen ions flow through, the ATP synthase will turn. So it's spinning. And while it's spinning, it's going to grab onto an ADP and a phosphate group, shove the two together, and make like 30 ATP. You get so many ATP out of this. So this is really our paying off phase, right? This is like you invest your money over time, and 30 years later, you're really feeling good about the amount of money that you've accumulated and all the interest and whatever that you got out of it, right? So this is really your payout phase. We did all this work earlier. Really, it's all for this last step almost. Okay. So why is it important for the electrons to go down the electron transport chain? So that we can pump the hydrogen. And why is the pumping of the hydrogen important? Is so that the hydrogen can naturally flow back through the ATP synthase. This is a passive transport. It does not use energy. It is facilitated diffusion because it's going through this molecule. And in fact, it's not only not using energy, it's making energy out of the process. The electrons will not go through the electron transport chain without oxygen bringing it over at the very end, the final electron acceptor. Okay, so this is the overall, all of the steps. You can pause right here and just take a look at how all 4 steps coordinate with each other one more time. Okay, now this is a good diagram for you to fill out. Let's take a look. We have glucose pyruvate, right? During this step, we are going to make 2 ATP. So this is ATP right here. We're also making 2 NADH right up here. And as you can see, it's going. NADH is going to the electron transport chain. Glycolysis takes place in the cytoplasm. Then we have pyruvate, 2 for every glucose, it says. pyruvate making acetyl-CoA 2 carbon molecule is going to make some CO2 along the way and some more NADH going to the electron transport chain. Then we have the citric acid cycle or the Krebs cycle, making CO2, ATP, and NADH and FADH2. And all those electron carriers are going to go to the electron transport chain. And our very last step, the electron transport chain, oxygen, right? We breathe the oxygen, we write oxygen in the equation. It's really important for us to have oxygen and use the oxygen because the oxygen is being used during the electron transport chain and forming water as a byproduct. We're not trying to form water. We're trying to use oxygen as the final electron acceptor, and we get 34 ATP. Over here is matrix, inner membrane. Okay, so here's our answer key. You're welcome to pause and take a look if you missed anything. Pyruvate processing over here, it says cytoplasm to matrix. The process itself really takes place in the matrix. We're just saying cytoplasm to matrix as in pyruvate needs to go from the cytoplasm to the matrix before it is processed. But technically, pyruvate processing takes place in the matrix. All right. One last look at our really important step, the electron transport chain. Step 1, NADH, FADH2 carry high energy electrons from all the steps, and the electrons are dropped off, as you can see. So this is words for you to pause and look at. Step 2, the electrons go down the electron transport chain and pump hydrogen now from the matrix to the intermembrane space through the inner membrane, right? You can see the inner membrane. So it's going through it. And then this is a low to high concentration active transport. So this is what allow us to build up this concentration of high concentration of hydrogen ion in the intermembrane space, the concentration gradient. Okay, so we got low concentration of hydrogen ions, and this also allows a negative charge, and the high concentration of hydrogen ions in the intermembrane space, and also a positive charge. And you can see oxygen is our final electron acceptor, combining with the electrons, forming water at the end. So this is really giving a strong, it's pulling, like really, it's like pulling, right? It's like if you were to have tug of war and you put a very, very strong person at the end of the line and it's really carrying the team. That's what oxygen is doing over here. Okay, moving on. Oh, to some answer key, apparently. So we have A through H, some questions and answers that you can go look at, but we've talked about all these different concepts already, so hopefully this makes sense. You're welcome to pause and take a look at the answers. And we did a crossword puzzle during class. You can take a look at these questions as well. You can pause and think about, do you know the answer? Okay, and here are the answers. All right, so that is all for our cell respiration review. If you have any questions, you can always come and find me. Good luck studying.